Hey, hey, hey. Welcome to a world of laughter, learning, and entertainment. Welcome to our channel, where today's focus is all about Gilbert Ling. Let's get started. Gilbert Ning Ling, December 26, 1919, November 10, 2019, was a Chinese-born American cell physiologist, biochemist, and scientific investigator. In 1944, Ling won the biology slot of the 6th Boxer Indemnity Scholarship, a nationwide competitive examination that allowed Chinese science and engineering students full scholarship to study in a United States university. In 1947, he co-developed the Gerard-Graham-Ling microelectrode, a device that allows scientists to more accurately measure the electrical potentials of living cells. In 1962, he proposed the association induction hypothesis, which claims to be unifying general theory of the living cell, and is an alternative and controversial hypothesis to the membrane and steady-state membrane pump theories, and 3 years later added the Polarized-Oriented Multilayer (PM or POM) theory of cell water. Ling carried out scientific experiments that attempted to disprove the accepted view of the cell as a membrane containing a number of pumps such as the sodium-potassium pump and the calcium pump and channels that engage in active transport. He died in November 2019, 1 month short of turning 100. Get ready to uncover the mysteries surrounding early life and education as we navigate its intriguing terrain. Ling was born in December 1919 in Nanjing, China. He grew up in Beijing and entered the National Central University Nanking University in Chongqing as a student of animal husbandry. After 2 years, he transferred to the biology department and received a biology BSc degree, minoring in physics and chemistry in 1943. In 1944, having done graduate work in biochemistry at the National Southwestern Associated University National Tsinghua University in Kunming, Ling won the 6th Boxer Indemnity Scholarship. In early 1946, he began his graduate study in the Department of Physiology at the University of Chicago under Professor Ralph W. Gerard. In 1948, he completed his PhD on the effects of metabolism, temperature and other factors on the membrane potential of single frog muscle fibers, which was published in December 1949 in a series of 4 papers in the Journal of Cellular and Comparative Physiology, Volume 34, Issue 3. He spent 2 more years under Professor Gerard as a Seymour Coleman postdoctoral fellow. Moving forward, we'll be taking a closer look at academic career. In 1944, Ling won the only biology slot of the 6th Nationwide Boxer Indemnity Fellowship to study physiology in the United States, which he took up in January 1946. From 1950 to 1953, Ling worked as an instructor at the Medical School of the Johns Hopkins University in Baltimore. His research and experiments led him to the conclusion that the mainstream membrane pump theory of the living cell was not correct. This early embryonic version of the association induction hypothesis was called Ling's fixed charge hypothesis (LFCH). From 1953 to 1957, he continued full-time research at the Neuropsychiatric Institute at the University of Illinois Medical School in Chicago. Beginning as an assistant professor, he was promoted 2 years later to tenured associate professorship. In 1957, he accepted the position of senior research scientist at the Basic Research Department of the newly founded Eastern Pennsylvania Psychiatric Institute. In 1962, his first book, entitled A Physical Theory of the Living State, The Association Induction Hypothesis, was published. At this time, Ling became director of a research laboratory at the Pennsylvania Hospital in Philadelphia. In 1984, Ling published his second book, In Search of the Physical Basis of Life. In October 1988, Ling's laboratory shut down due to his inability to attain research funds from National Institutes of Health and other funding agencies. Raymond van Damodian offered to support him and 2 of his staff, Margaret Xenfeld and Dr. Zhendong Chen. From 1982 to 1985, he was a co-editor-in-chief of the Physiological Chemistry and Physics and Medical NMR Journal, and after 1986 served as sole editor-in-chief. In 1992, Ling published his third book, A Revolution in the Physiology of the Living Cell. In 2001, his fourth book, Life at the Cell and Below Cell Level, was published and subsequently translated into Russian and Chinese. In 2011, his wife of 60 years, Shirley Wang Ling, died of pancreatic cancer. In 2014, at the age of 94, he published his fifth book, What is Life Answered, a response to Erwin Schrödinger's 1944 book What is Life. He published over 200 scientific papers, although much of his later work was largely ignored by the scientific community. In this chapter, we'll be unraveling the enigma of Gerard-Graham-Ling microelectrode and discovering its transformative power. Also known as the Ling-Gerard microelectrode, and after the ERS further developed into the glass capillary microelectrode, has played a vital role in modern neurophysiology and medicine. John Eccles applied the microelectrode to studies of activity of individual units within the spinal cord and brain, and Andrew Huxley used it in muscle cells. In 1963, Hodgkin with Huxley won the Nobel Prize in Physiology or Medicine for their work on the basis of nerve action potentials, the electrical impulses which enable the activity of an organism to be coordinated by a central nervous system. Hodgkin and Huxley shared the prize that year with John Eccles, who was cited for his research on synapses. Worldwide use of this new microelectrode spread rapidly after this and has subsequently proven to be one of the most important devices applied to the study of cellular physiology. The microelectrode in use today is essentially the same as this, except that it usually contains a concentrated salt solution and is commonly referred to as the glass capillary. In 1950, Gerard was nominated for the Nobel Prize for helping to develop the microelectrode as used in electrophysiology. Get ready for a thought-provoking discussion as we delve into association induction hypothesis and its impact on our understanding. An alternative and controversial hypothesis to the membrane and membrane pump theories, the association induction hypothesis is a claim related to the properties and activities of microscopic assemblies of molecules, atoms, ions, and electrons of the smallest unit of life called nanoprotoplasm. Ling wrote books describing his hypothesis in 1962 and 1984, and later self-published other books. In this section, we'll be deep diving into polarized-oriented multilayer theory, unraveling its complexities and uncovering valuable insights. In 1965, Ling added his polarized-oriented multilayer of cell water to the association induction hypothesis. The theory argues that intracellular water is polarized and oriented and thus dynamically structured. Brace yourself for a captivating discussion on criticism as we explore its nuances and implications. In 1974, Lawrence G. Palmer and Jagdish Gilati tested one aspect of Ling's theories, namely, whether potassium ions within the cell are bound or free. Contrary to Ling's prediction, they found that in fact potassium ions within frog skeletal muscle cells are free. With that being said, let's now move on to publications. Gilbert Ning, A Physical Theory of the Living State, the Association Induction Hypothesis. Lacedal Publishing Company, a division of Random House, Inc. London, 1962. 682 pages. Library of Congress Catalogue Number 60-11835. Gilbert Ning, In Search of the Physical Basis of Life. Plenum Press, New York and London, 1984. 791 pages. Gilbert Ning, A Revolution in the Physiology of the Living Cell. Krieger Publishing Company, Malibu, Florida, 1992. 378 pages. Gilbert Ning, Life at the Cell and Below Cell Level, The Hidden History of a Fundamental Revolution in Biology. New York Pacific Press, 2001. 373 pages. Gilbert Ning, What is Life Answered? Cushing Malloy Inc. Ann Arbor, Michigan, 2013. 120 pages. Leave a comment below and let me know what you thought of this video.